Hello ma'am, welcome to our presentation video. Our presentation video topic is DNA sequencing. We have two group members. I am Sifat, my ID is 121152500, and my another group member is Mr. Jahan Tania. Our group ID is 121152389. Let's see our presentation video. And now I will show you the DNA sequencing process. First, the target DNA is from the human, fish, frog, etc. is broken up into small pieces and packed into small loops of bacterial DNA known as plasmids, and this is the plasmid. As the bacterial cells grow and divide, they replicate the plasmid and target DNA as well as their own DNA. In preparation of sequencing, the plasmid containing the target DNA must be separated from the rest of the bacterial cell. Robots add detergent to break open the bacterial cell and release the DNA. The samples are placed in a centrifuge to spin. This process gathers the heavy components of the cell at the bottom of the tube. Only the plasmid DNA carrying out target sequencing is left in solution. This plasmid DNA is filtered and washed to further purify the sample. Here you see the plasmid in solution. This is the bacterial components. This is the zoom picture. The solution containing the plasmids with target DNA is transferred to another plate. And the chemical required to start the sequencing reaction are added. The sequencing mixture of plasmids and target DNA nucleotides, fluorescently labeled nucleotides, enzyme and primer sequences to get the reaction started is then put onto a heating block. You see the enzyme, nucleotide, primer, plasmid, and these are fluorescently labeled nucleotides. Let's see how these ingredients combine in the Sanger sequencing reaction. The reaction is initiated at either end of the target DNA while it joins with plasmid DNA. It's easier to see the action if we show the DNA unwound. First, the DNA is heated so that its two DNA strands separate. The temperature is then lowered so that a short length of DNA sequence, a primer that is complementary to the known plasmid DNA sequence, can bind. Finally, the temperature is raised slightly to enable the enzyme to bind to the short section of double started DNA. The enzyme then starts to make a new strand of DNA by incorporating free nucleotides that are complementary to the target DNA. The enzyme will continue to extend the strand until it randomly incorporates a fluorescently labeled nucleotide. The fluorescently labeled nucleotides are chemically altered so they terminate the DNA strand and the enzyme pulls away. The cycle of heating and cooling is repeated many times, generating a large number of fragments of different lengths that end in fluorescently labeled paste. At this stage, the fragments are floating freely in one of many tiny test tubes in a plate known as wells. The sort of the fragments out by size and detect which bases have been added when the plate is loaded into a sequencing machine. Inside the machine, the samples are transferred into the glass capillaries, where an electrical charge discharges the negatively charged DNA molecules through a gate matrix. As they move, the longer DNA fragments are slowed down more by the gate than the shorter fragments. So when all the DNA fragments from our sequencing reaction are run through the capillary, they are sorted by size, from shortest to largest. And a laser at the end of the capillary is used to excite the final fluorescent base, which is recorded as a colored peak of Burr. This process means that the fragments that incorporated fluorescent tags and terminated early will be read first. The laser can detect only the fluorescently labeled base at the end of each fragment. So each colored bar represents the final base of each strand of DNA that was made in the sequencing reaction, ordered from shortest to longest. Capillary sequences can be used for many samples at a time. This output shows 96 samples as rows and can read the equivalent of 500 to 800 DNA letters from each sample. Thank you, ma'am. Please.